Of the cytology 2. You remember last time on Friday we have said eukaryotic cells could be said to have a cell membrane, protein, and the nucleus. Cell membrane, protoplasm, and the nucleus. Today I'm going to describe about protoplasm, which has cytoplasm or cytosol, organelles, and inclusions. So I'm going to take you through each one of these, one after the other. If we begin with the cytoplasm, which I don't say much about it because there is no much structure to be seen for histology, because it is made by proteins, electrolytes, and metabolites dissolved in water, which makes about 75 to 90% of the cytoplasm is water. And this contains proteins, electrolytes, and metabolites fitted in between the cytoskeletal system of the cell. The cytoplasm could be divided regionally into two. One is the inner region and another one is the outer region. If we look at this picture here, we can see color coded. The inner one is more of the greenish colored region, where we have many small particles moving randomly in every direction and getting bombarded one with another, in addition to which we have specified directed movements of vesicles and organelles. So many of the organelles and the vesicles are found in the inner aspect of the cytoplasm, where we have directional movements of these organelles and the vesicles as well. But when we look at the periphery of the cell, which is here indicated by the reddish yellowish outline, consists more of supporting microfilaments. These are mainly for maintenance of the cell shape and movements of the cell and supporting. So that's what we can see with regard to the general outline of the interior of the cell, which could be divided into an outer region and an inner region. So many of the organelles we are going to speak now in the subsequent slides are found mainly in the inner region of the cell. When we look at the next part of the cell, which we divided it as organelles, these are plenty of small structures for specific functions. You can say they are small factories. There are plenty of such small factories specializing to carry on different functions of the cell. Based on the requirements of these organelles to have its own room or not, we can divide them into those which are surrounded by membrane to have their own specific environment as membranous organelles, which are making those listed here. And we also have other organelles which have no specialized environment, so they are not surrounded by their own membrane. So I'm going now first to describe for you the different membranous organelles, to be followed by those with no membrane. So let me now begin the first membranous organelle. By the way, are you with me? Are you with me? Okay, now, all right. Okay, I see. So I will go ahead. So the first membrane-bound organelles in the list are endoplasmic reticulum. This endoplasmic reticulum are made by network of anastomosing channels within the cell. These are interconnecting one with another, like this one, and like this one here. These are making channels, tubular structures, which are branching and anastomosing one with another. And they produce substances which would be wrapped by membranes that would be pinched off from these tubular structures and be transported to next organelles. Organelles interact. Substance such as proteins would be produced by one given organelles and would be taken, would be transported to another organelles. So they are interacting. So this endoplasmic reticulum, which are made by anastomosing channels of tubular structures of various sizes, are making substances which would be wrapped by membrane and form transfer vesicles. These are to be transported to the next organelle, which happen to be Golgi apparatus. Based on presence or absence of another organelles known as ribosomes attached on the surface of this endoplasmic reticulum, we can divide the endoplasmic reticulum into rough or granular endoplasmic reticulum, which have the ribosomes attached on their wall, and the smooth or agranular endoplasmic reticulum, which have no attached ribosomes on them. So there are two types of endoplasmic reticulum. One is rough, another one is smooth. When we look at the rough endoplasmic reticulum, it looks like this one, or this is electron micrograph of endoplasmic reticulum, real picture. This one on the left is a diagram. You can see on the surface of this rough endoplasmic reticulum, there are small granules. These granules are granulated bodies are representing the ribosomes. This rough endoplasmic reticulum are connected, or their membranes are continuous with the outer membrane of the nucleus. When we reach at the stage of looking at the nucleus, we will see that the nucleus is surrounded by double membrane. I'm not saying the lipid bilayer of one membrane, but the nucleus is surrounded by two membranes. One is inner, another one is outer. This outer nuclear membrane, as you could see here, this is a nucleus. This outer nuclear membrane is continuous with the rough endoplasmic reticulum membrane. And on this rough endoplasmic reticulum, same as on the outer membrane of the nuclear membrane, there are ribosomes attached because they have receptors. There are receptors for this ribosomes made by docking proteins, as well as glycoproteins, riboforins, which would hold on the ribosomes on their membrane. This, therefore, make the ribosomes fixed on their outer surface, and this plasmic reticulum specializes in production of proteins which would be taken to the Golgi apparatus, modified, and be exported or integrated into plasma membrane, as well as make the several enzymes of another organelles known as the lysosomes. So the rough endoplasmic reticulum, under the instruction of the nucleus, produces proteins which are for export, for integral proteins of membrane, as well as for those making lysosomal enzymes. That's the specialty of rough endoplasmic reticulum. So this can tell you that cells which are specializing, for example, for secretion of some substances, such as, for example, glandular cells, need to have large amount of rough endoplasmic reticulum. This is what the rough endoplasmic reticulum look like and does. On the other hand, the smooth endoplasmic reticulum, which are found just next to the rough endoplasmic reticulum, in some cases even they are in continuity with each other, and they look like this one. Here in this diagram, you can see on the right side of this endoplasmic reticulum, the membrane is smooth, whereas those found on the left side, as I'm showing here with the pointer, if you are following, these ones are rough, but these ones do not have the granules, so they are named as smooth or agranular. A means non-granular endoplasmic reticulum. When you compare the smooth with the rough, the smooth endoplasmic reticulum have more of a tubular and vesicular type of nature, so that they are wider, they have wider lumen. The lumen is known as cisterna or cisterne, plural. So the cisterne of the smooth endoplasmic reticulum, as are indicated here in the right side of the picture, are wider, here indicated by the blue outline, are wider compared with those of the rough endoplasmic reticulum. They do not have ribosomes, so they are not involved in the production of protein. You know, ribosomes are for production of proteins. So the smooth endoplasmic reticulum do not have organelles known as ribosomes attached to them, so they are not involved in the protein synthesis. Instead, they are synthesizing steroid hormones. They are also important for lipid metabolism, glycogen breakdown, as well as detoxification. In addition to such important functions in different types of cells, the smooth endoplasmic reticulum are also involved in calcium storage, specifically in skeletal and cardiac muscles. You know, calcium ions are important for muscle contraction. When muscles are not contracting, the calcium ions should not be found nearby the contractile elements of the muscle. Instead, they are stored in the smooth endoplasmic reticulum. And when the muscles are needed to be contracted, these calcium ions should be released. So the smooth endoplasmic reticulum are like storage sites for calcium ions, and they are very well formed in such muscle cells. As a result, they are named as sarcoplasmic reticulum. I'm saying the sarcoplasmic reticulum of muscle cells are nothing but the smooth endoplasmic reticulum, and these are found in two of the three types of muscle cells. The third type of muscle cells, known as the smooth muscle, do not have this type of nature, because the smooth endoplasmic reticulum and smooth muscles are not for storage of calcium ions. As a result, we don't have sarcoplasmic reticulum in smooth muscle cells, but we can have, in fact, we have smooth endoplasmic reticulum in smooth muscles for other purposes. So you see again, the smooth endoplasmic reticulum are well formed in cells such as skeletal, cardiac muscles, in cells such as, for example, liver cells, which are specializing for detoxification of substances. These are also very well formed in steroid and lipid synthesizing type of cells. These are where they are found, and this is what I can tell you briefly about the smooth endoplasmic reticulum, which are same as rough endoplasmic reticulum, made by channels of tubular structures, which are branching and anastomosing, but they do not have ribosomes, and they have different function when you compare them with the rough endoplasmic reticulum. Then the next organelles I would describe to you would be the Golgi complex, which are also known as Golgi bodies and the Golgi apparatus. This Golgi complex are again made by membrane channels. These are all membrane-bound organelles, mind you, I'm describing now the first part of my lecture. So same as endoplasmic reticulum, Golgi complex are also formed by membrane channels. This time the channels are not the type of tubular structures, unlike in the endoplasmic reticulum. Instead, they are flattened and sac or bag-like structures, like this one. This is what the Golgi apparatus looks like. It is made by transversely running, flattened, sac-like membranes, which again have cisternae. Cisternae means nothing but the interior, the space within the membrane. So we have here again, the channels are anastomosing, but they are not tubular, they are flattened, sac-like channels made by about 3 to 10 in number. These are piled one upon another, and they are located near the rough endoplasmic reticulum. I told you the rough endoplasmic reticulum is found nearer to the nucleus. Not only nearer, but it is even connected with the outer membrane of the nucleus. So the rough endoplasmic reticulum is found nearer to the nucleus. The Golgi apparatus is also found nearer to the nucleus, around the cell center. The nucleus is found around the center, nearer to the centrosomes, around the centrioles. So this is where they are located. And this Golgi apparatus would receive the transfer vesicles from both rough and smooth endoplasmic reticulum. Once substances, proteins, steroids are produced in the endoplasmic reticulum, they would be wrapped by membrane, forming transfer vesicles like here, which are transported towards the Golgi complex. So this Golgi complex has a surface which is facing towards the nucleus, which is receiving transverse vesicles. And this surface of the Golgi apparatus is named as forming convex or entry or outer and a cis surface, because this is the region where the vesicles are coming in and joining the membrane, like this one. It is receiving surface of the Golgi apparatus. The opposite surface is the surface through which, once substances received are modified, will be released. So this opposing surface is named as maturing, condensing, exit, or other. It has several names. Surface. Usually this surface is concave, whereas the opposite surface, the receiving surface, is convex. So through the maturing surface, condensing vacuoles or vesicles will be released out. This will be transported towards the plasma membrane and be released out, or will be released as newly formed organelles, such as the lysosomes, or they will be integrated into the plasma membrane. So this Golgi apparatus has therefore two surfaces, receiving surface and an outer surface. why does this Golgi apparatus receive and then afterward sends? It is because it also has the function of synthesizing, especially carbohydrates, polysaccharides, to be added onto proteins and steroids and other substances which it receives from the endoplasmic reticulum, like this one here. In this diagram, you can see it receives, and the green outlines are the interior of the Golgi apparatus, where carbohydrates, for example, are produced. And these carbohydrates are added to proteins and lipids, forming glycoproteins, proteoglycans, as well as glycolipids. And this means that the substances received from the endoplasmic reticulum are modified, are added with polysaccharides. And the Golgi apparatus also sulfates, such as, for example, glycosaminoglycans are sulfated. And these are therefore get modified. And finally, the modified substance are packed. These are packed and concentrated and then released towards the cytoplasm, in which the vesicles are found dispersed and move as are desired. If these are to be the lysosomes, they will remain within the cell. If these are to be part of the membrane protein, they will move towards the plasma membrane and get integrated. If they are to be released out as secretory product, they would be released. So that's the function of the Golgi apparatus. Literally, its function is modification of secretory products received from the endoplasmic reticulum, modifies them, packs them, and then dispense them. It is like the post office. It is like you would get your items purchased from shops, would be packed, and then be handed to you. So this is what the Golgi complex does. This is the second membranous organelles. So the next one is the lysosomes. Before I move to the lysosomes, are you with me? Are you following? Do you have anything to ask? Okay, I see. Okay, thank you very much. Thank you. Okay, I would move on. So the next organelles I would describe for you would be the lysosomes, which partly I've been telling you now and then that it has enzymes which are produced by rough endoplasmic reticulum and gets modified by the Golgi complex. Yes, indeed, lysosome is small, spherical, electron-dense bodies which contain about 40 specific enzymes which belong to the hydrolytic group of enzymes, which are specializing to hydrolyze chemical bonds, breaking down bigger particles into smaller molecules, such as, for example, the nutrients received by the cells or anything that are taken from outside would come to the lysosomes, and these lysosomes would fuse with the vesicles, whereby the content of the vesicles would be internalized into the lysosomes. And within the membrane-bound lysosomes, digestion would take place. And out of such digestion, substances that are required to the cells would be pumped out into the cytoplasm, whereas those which are undigested or not needed by the cytoplasm would remain within the lysosomes, and these lysosomes would gradually get aged. When this would be aged, would be released out of the cell or would remain within the cells as debris, making one type of inclusions. The reason why these lysosomes are needed to be bound by membrane is, for example, look at the pH required for the lysosome is acidic. It is about 5, whereas the cytoplasmic environment is about neutral pH, about 7. And in addition, some of these enzymes are able to digest the other organelles. They are able to digest and destroy the cytoplasm. So because of this, these enzymes should be bound and separated from the rest of the organelles by their own membrane. These lysosomes can even digest self. When the lysosomes break down, if the membrane is broken down, these enzymes would leak out into the cytoplasm and able to digest the cell itself. And as a result, these lysosomes are known as suicide packets. Suicide packets. These lysosomes could also be released into outer aspect of the cell to digest substances found outside the cell, such as, for example, extracellular matrix could be destroyed if needed by the enzymes of lysosomes. So these lysosomes are able to kill the cell itself or anything of the cell itself. These lysosomes, as I have been already describing to you now and then, are synthesized by initially the lysosomes, the endoplasmic reticulum, and then get modified, glycosylated within the Golgi apparatus and be released out into the protoplasm. So that's what I can tell you about the lysosomes. The next organelles, which are comparable with the lysosomes but bigger and have different types of enzymes, are peroxisomes. Peroxisomes contain again about same number of enzymes, but these enzymes are different. These are oxidative type of enzymes which are able to produce hydrogen peroxide. This hydrogen peroxide is toxic to microorganisms, and it is important for detoxification of toxic agents. So cells that are required to detoxify, again, cells such as liver cells, need to have large amount of this peroxisomes or microbodies because they are able to produce hydrogen peroxide because they are oxidative type of enzymes. Their ability to produce hydrogen peroxide specializes them to be involved in detoxification. And the enzymes we have in these peroxisomes are synthesized by 3 ribosomes. I'm going to mention to you that we have 2 types of ribosomes. Because of their location, they are specializing in production of different types of proteins. And those which are attached to the rough endoplasmic reticulum are specializing proteins to be released outside or also produce lysosomes. Whereas ribosomes, which are not attached to rough endoplasmic reticulum, produces proteins for within the cell use, including, like, for example, proteins or enzymes of peroxisomes. So peroxisomes are not produced by rough endoplasmic reticulum. Instead, they are produced by the 3 polyribosomes we are going to describe soon. These peroxisomes are able to break down and get multiplied. You know, other organelles are synthesized from scratch, but such peroxisomes would able to grow. Proteins needed, these enzymes needed, would be produced by 3 polyribosomes and make the peroxisomes grow and then split. And they multiply by a process known as fission. We call this type of replication fission. So they are able to break down for purpose of multiplication. Then we have another organelles in the list, the mitochondria. You know, mitochondria are important for supplying the energy currency of the cell, which is ATP. So the mitochondria are specializing for production of ATP, and the cells which are active and doing important activities or engaged in activities. The activities could be production of secretory products, maybe cell division, maybe cell motility, or any type of activity may require the cell to have large amount of energy. So in this type of cases, the cell need to have large amount of mitochondria. When cells are resting and are relatively inactive, the number and the type of mitochondria would be less, and they are smaller in size. So because of this, they are larger in most cells, spherical, filamentous, or rod shape. So they are variable in shape as well as sizes, and they can be taken as the largest organelles within the cell. They can also be multiple or smaller in number. And they are again membrane bound. This time their membrane is like the nuclear membrane I told you, come as double. So here, look at this. This is a cut part of the mitochondrion. And here you can see one outer layer and another is an inner layer. Each one of this is made by the lipid bilayer. I'm not speaking about the lipid bilayer or the leaflets, the double leaflets of the phospholipid. Instead, each one of this is made by the phospholipid bilayer, but comes as duplicates. It is therefore made by an outer membrane and an inner membrane. When we look at the outer membrane, it is smooth throughout. It doesn't fold. If it is spherical, it just remains spherical. If it is rod-shaped, it is just remaining as a rod shape. And it is porous. Substance transportation across this outer membrane is faster. But when it comes to the inner membrane is thrown into folds, inward projections to increase surface area. So it has some folds which may be tubular or shelf-like, and this type of foldings are named as cristae. Crista is singular. Cristae, when you add E, is plural. So here you have the cristae look like this. The cristae in many of the mitochondria, in many cells, these are shelf-like, whereas in cells which are specifically producing steroid hormones, the cristae are tubular, like this one. These are tubular, these ones are flattened and shelf-like. So by looking at the shape and nature of the cristae, you can tell the activities of the cells. Many of the cases, many cells have shelf-like, plenty, whereas in steroid-producing cells, the cristae are tubular and even much more formed. And this mitochondria also are able to, or just not able, but just split. They are able to fuse as well as split. Two mitochondria can come to form one, and when they need to be multiplied, they just simply grow and split. Because of the foldings of the inner mitochondrial membrane, we have two spaces, two compartments within the mitochondria. One compartment is the outer, which is found between outer membrane and the inner membrane. We call this outer chamber or intermembrane space. Inter means between. You see, when we say in anatomy, inter, look at this E. Inter, it is referring to between. If this one was intra, it means within, inside. Like, for example, this space, which is intermembranous space, is running deep into the folds of the inner membrane. It is running deep into the crystal. So this space is also known as intracrystal space. So it is intracrystal space. It is a space found within the crystal. It is not found between the crystal, like this one is named as intercrystal space. Indeed, the next space, the inner compartment, this compartment is found between adjacent crystal, so you can call it intercrystal space. So intercrystal space, it is also known as the matrix. This matrix is the biggest, and it contains several enzymes, water, solute, granules, as well as calcium and magnesium ions. Not only this, the inner space, the matrix, also contains ribosomes, RNA, and more interestingly, it also contains DNA. Normally, in the cell, eukaryotic cells have their DNA located within the nucleus. But we also have a small amount of DNA. Small genes are also present within the mitochondria. We call this mitochondrial DNA. They are like the DNA of prokaryotic cells. They are spherical, they are circular, they are not linear. The nuclear DNA are thread-like, they are linear, whereas the DNA of the mitochondria, like those found in the prokaryotic cells, are spherical. And they produce RNA for transcription of small amount of proteins. These proteins they are producing are not even enough for the mitochondrial demand. So they are producing small amount of proteins for the mitochondria. This DNA are different with regard to their inheritance from those of the nuclear DNA. The nuclear DNA are inherited from both paternal and maternal, meaning the DNA of the nucleus come from both sperm and the egg. The mitochondrial DNA, on the other hand, is said to be inherited only from the oocyte. So this DNA are all maternal DNA. So that's what I can tell you about the mitochondria. And as I've said already, the function of the mitochondria is production of ATP. ATP required for the energy supply of the cell. So from structural point of view, that's what I can tell you. From a biological aspect, you would see this mitochondria very much in detail in other disciplines such as the biochemistry. So that's what we can say about mitochondria. And the next organelles, important machineries, membrane bound and found within the cells, engaged in activities, active ones, are the endosomes and the phagosomes. We already mentioned when we looked at the membrane. When we looked at the membrane, I've shown you these pictures here that substances are internalized into the cell, being wrapped by membrane, forming vesicles. So these are making the endosomes and the phagosomes. These endosomes and the phagosomes may be to cross the cell. In that case, we name them specifically for the endosomes, transcytosis, but this may also be destined for intracellular digestion. And whatever is the case, they are engaged in some types of activities. So in this type of manner, they are active and they belong also to the active structures within the cells, which are nothing but organelles. And this are maybe to be digested. In case of phagosomes, they may be heterophagosomes or autophagosomes. These heterophagosomes are those which are internalized from outside. But we can also have part of the cell itself be wrapped by a membrane and make what are named as autophagosomes. The auto is indicating self. Why do we have such autophagosomes? Maybe these are to be destroyed, maybe to be digested, maybe they are not needed, so that they are now wrapped by membrane and they are part of the cell itself, such as, for example, if part of the rough endoplasmic reticulum is aged or is required to be digested, it would be pinched off, surrounded by a membrane, and it would be taken to the lysosome. So these are just nothing but, again, phagosomes. They are, however, self, so we call them auto, in contrast to the heterophagosomes internalized from outside. And when they are coming to fuse with the lysosomes, they make now a fusion with the lysosomes and be named as heterophagolysosomes or autophagolysosomes. Within the lysosomal membrane, these are to be digested and their fate is now finished. So these are also membrane-bound structures, important structures within the cells. And this has now brought us to finish about membrane-bound organelles. I listed to you the membrane-bound organelles, includes the endoplasmic reticulum, the Golgi complex, the lysosomes, the mitochondria, the peroxisomes, and the endosomes. And many of these interact with each other. They interact not only with each other, they are also interacting with the nucleus. For example, the instruction for production of protein by the endoplasmic reticulum, rough endoplasmic reticulum, comes from the nucleus. And the outer nuclear membrane is in continuity with the membrane of rough endoplasmic reticulum. So the nucleus also interacts with them. Not only this one, the vesicles they produced would ultimately be interacting with the plasma membrane. So those organelles which are interacting with each other are named as endomembrane system. This includes all membrane-bound organelles except peroxisomes and the mitochondria. These do not interact with the other organelles I mentioned, as well as with the plasma membrane. So except these two, the membrane-bound organelles, along with the nucleus and the plasma membrane, make what is named as endomembrane system. Therefore, this is what I can tell you about the important organelles which require specific and unique environment for themselves, and as a result, wrapped by their own membranes. And we call this membrane-bound organelles, which are different from those which are found free within the cytoplasm, and these are named as non-membrane-bound organelles. So before I move to begin discussing about these non-membrane-bound organelles, do you have anything to ask, or are you comfortable with me? Are you following? And shall I move on? Okay, I see. Thank you, you are listening. So, okay, thank you very much. Thank you. Okay, so let me move down and maybe later we can chat. So let me at least describe to you one of the non-membrane bound organelles, and then we may take a few minutes break and come back for another discussion. And that time we can also have a chat. The non-membrane-bound organelles include the ribosomes for making proteins. I've been mentioning this already. And then we have different types of filamentous proteins, which are collectively named as cytoskeleton of the cell, and the centrioles located at the cell center. So these are the organelles which are found freely within the cytoplasm. So let's begin with the ribosomes. These ribosomes are made biochemically from several types of ribosomal RNA. They are transcribed by the nuclear DNA, and there are different types. These are linked with specific ribosomal proteins. These proteins are made in the cytoplasm under the instruction of the nuclear DNA, and then they would be taken into the nucleus. And the nucleus, there is a region where ribosomes are assembled or synthesized. So in this region of the nucleus, which is named as nucleolus, we name it nucleolus. I didn't say nucleus. Nucleus and nucleolus are different. Nucleolus is a region within the nucleus where the ribosomal RNA are transcribed and get assembled with the specific ribosomal proteins and form ribosomes to be released out of the nucleus into the cytoplasm. So that's how the ribosomes are produced. When these ribosomes are produced, they are produced with two subunits. Here they are indicated as this in this diagrams. Here you can see a small subunit and a bigger subunit. We call this large and small subunits. They would assemble together to give us a ribosome. So this is a ribosome, this is a ribosome, here is another ribosome. So these are discrete bodies, separate entities, which are ribosomes found within the cytoplasm. These ribosomes may be found dispersed within cytoplasm with no activity. So we call them free ribosomes. But when they are to be engaged in activities, they would attach to a single messenger RNA like this one. This green outline is to indicate a messenger RNA. Onto this messenger RNA, look at this, there are plenty of ribosomes which have now got attached to it. When they are assembled, attached to a single strand of messenger RNA, we call them polyribosomes or polysomes. So this is a polysome which is now able to produce protein. This type of polyribosomes are those which are able to produce ribosomes by assembling amino acids into proteins. So you see, the ribosomes we have in a cell may be found as free ribosomes, not active, or polyribosomes or polysomes, which are active because they are attached to messenger RNA. Let's now look at those which are active. Let's look at these polyribosomes, how they are found. They themselves are found in two manners, in two ways. One way they are found is freely within the cytoplasm, like this one. We name them as free polyribosomes, free polysomes. They are those which are synthesizing proteins. I told you, for example, for peroxisomes, proteins for intracellular use, proteins for organelles and structural elements found within the cells. except the lysosomes. So these are named as free polyribosomes. On the other hand, another group of polyribosomes are attached on the outer surface of the nuclear membrane as well as the rough endoplasmic reticulum. This diagram here is showing part of a rough endoplasmic reticulum. Look at this is the membrane of the rough endoplasmic reticulum. On it, here we have the polyribosomes attached to it. So this type of endoplasmic reticulum is now named as the granular type of endoplasmic reticulum. So you see, when I told you that earlier we have two types of endoplasmic reticulum, one has attached ribosomes. I was referring to the polysomes or polyribosomes, which are attached to it. So this is what rough endoplasmic reticulum looks like. Again, these are able to produce specific type of proteins. Such produced proteins are specifically for export, for release out of the cell, or to be integrated into plasma membrane, or to make specifically the enzymes of lysosomes. So we have in this type of manner, we have two types of polyribosomes. One are found freely within cytoplasm. They are active. They are engaged in production of proteins, but these proteins remain within the cell. Whereas the second type of polyribosomes are to be released out of the cell. So again, this type of their specific localization can tell you that all cells do not have same amount and same type of organelles, like glandular cells, like neural cells, which are known to produce several substances to be released outside. Glandular cells are producing secretory products, enzymes to be released out of them. So they need to have the attached type of polyribosomes, meaning they are having large amount of rough endoplasmic reticulum. On the other hand, those cells which are not needed to secrete outside, but maybe they are required to produce large amount of proteins for local intracellular use, they need to have a large amount of free polyribosomes. This type of cells may even be dividing cells. Cells, before they divide, they need to have large amount of organelles, which are going to split into two daughter cells. So they need to have a large amount of proteins to be produced. In this type of cases, they need to have large amount of polyribosomes. So you see, this can tell us the reason why we should not think that organelles are all found in a similar type of manner throughout the different types of cells. As a result, you see, the ribosomes, like this one, are found in cells such as this are neurons. They are found as plenty. And under light microscopy, you cannot distinctly identify organelles, I told you last time. But when they are plenty, you can see them granulated. You can see them stained, depending on the type of staining. And as a result of this, even before clearly they were discovered that rough endoplasmic reticulum are found plenty in the neurons, they were discovered or they were known to have, neurons were known to have large amount of granules. And these granules were named as little bodies. So neurons have a large amount of Nisl bodies, but later these Nisl bodies have been discovered to be rough endoplasmic reticulum. Same is true for glandular cells. Even before they were discovered that these are rough endoplasmic reticulum, they were named as ergastoplasm. So ergastoplasm are granulated bodies identified in many secretory cells, glandular cells, as course granules, but later this also have been discovered as rough endoplasmic reticulum, nothing but the polyribosomes attached to endoplasmic reticulum. The name remained. So in all the texts, you can read in glandular cells, there are plenty of ergastoplasm. In case of neurons, including recent books, neurons are named or are known, rather are known to have large amount of nestled bodies. These are just nothing but rough endoplasmic reticulum. Why do we have large amount of rough endoplasmic reticulum in the neurons? Is because neurons communicate by chemical messenger molecules. We call this neurotransmitters. So one neuron to stimulate another neuron, or to stimulate or inhibit, or anyway to communicate with another cell, they are releasing substances. These substances are largely chemical transmitter substances, many of which are known to be polypeptides. So because neurons need to produce plenty of polypeptides to be released outside, they need to have large amount of well-formed polyribosomes and rough endoplasmic reticulum. So that's what I can tell you about the ribosomes and the polyribosomes, which are free non-membrane bound organelles found within the cells. And before we just move on to look at other organelles, I suggest we need to have a few minutes break. But if you wish to have some question, feel free to ask. You may chat or you can ask. Otherwise, I'm going to dismiss the class for a few minutes. Now it is almost 10, so class will resume at 10:30. Right at 10:30, we can meet. So there is a question here. Can we get a specification for our exam regarding the limits that we should refer? So just with regard to the exam, I am going to follow the request from the module coordinator. So it all, I guess, would be MCQ types, multiple choice type of questions from what we covered. And I have the right to move up or down from the books. I told you have to enrich this. But the basic things have already been covered. More than 70% are all covered. So you have to concentrate on the PowerPoints, but you have to enrich it. It is just obvious. Any lecture is not exhaustive, so you have to enrich it. But more than 60–70%, I feel I have covered. I already have covered many things, so you have to concentrate and revolve around the PowerPoints. So what is fission and also autophagosomes? Fission is just a splitting. It is a sexual type of reproduction in cells. In some cells, prokaryotic cells like bacteria, they replicate their materials, whatever they have, including their DNA, and without having nucleus, without having mitotic spindle, they just simply split. So for some organelles, such as peroxisomes, before they split, they grow, and then they split. They divide into halves. In case of mitochondria, they also grow, including their DNA. Their DNA also replicate, but it's not like the nuclear DNA we know, which should be aligned at the equatorial plates and then divide. It's not like that. So in the case of mitochondria, both the maternal DNA as well as the other proteins would grow and then divide. Autophagosomes is just self-bodies, self-organelles, aged ones, those which are required to be digested would be wrapped by a membrane, creating autophagosomes. What special factor distinguish or make organelles to be membrane bound? Or the requirement? Why do you need to have a bedroom or a kitchen or living room? The reason why you have in your building separate rooms is because you need to have it for special purposes, which should be done privately there. So here also, membrane-bound organelles need to have their own membrane. So any lysosome. What is fission and okay, I did. Lysosomes are enzyme wrapped by membrane. Phagosomes, you can say, aged organelles to be digested. So phagosomes, autophagosomes are to be digested. Phagosomes are membrane-bound substances to be digested. Lysosomes are enzymes surrounded by membrane for digestion. What the vision I did. No, it is mitochondria and peroxisomes that are dividing by fission. It's not lysosome. It is peroxisome and the mitochondria. Normal. Yes, that's it. Why not this? Why is this not possible? Fission is just a division. You just break a chalk into half. That's what fission is. To grow, the proteins are synthesized by cytoplasmic polyribosomes and be integrated into the peroxisomes. And then they grow and they divide. And for mitochondria as well, many of the proteins making part of the mitochondria, the enzymes found in the mitochondria, the membranes of mitochondria, are not produced by mitochondrial DNA. I told you, the mitochondrial DNA produces only a limited amount of proteins for the mitochondria. Otherwise, many of the proteins are to be produced from outside. The mitochondria of the DNA happened to be the maternal because the oocyte has mitochondria, whereas the sperm cells do not, the mitochondria of sperm cells do not get into the oocyte, so they remain outside. They are only required for sperm cells able to swim and able to fertilize. At the time of fertilization, the sperm cells do not come with organelles. They only come with their DNA material. The organelles of the zygote, within the zygote, there are plenty of organelles. These are to be produced under the instruction of the nuclear DNA, which the nucleus of the DNA comes from both paternal and maternal. They are going to produce proteins for all organelles, including for the mitochondria. But within the mitochondria, the DNA is just already there. It is coming from the oocyte. It is not the sperm cell that will come with this DNA. So this DNA is maternal. It is not paternal. That's the reason. It's so far, I just simply told you only mitochondria has double membrane. Next day, I will tell you the nucleus would also have double membrane. as the different parts of cell have different types and number of ribosomes. I don't understand that. As different parts of the cell have different types and number of yes, they all have different amount of organelles. The specific, these are all found in all cells, but the amount size differs. Endosomes are usually for pinocytosis. Phagosomes are usually for phagocytosis. This one would much become clear to you in biochemistry, but what I can tell you today would be because of the enzymes of peroxisomes, hydrogen peroxide needed for detoxification, as well as killing microorganisms, are produced by the enzymes of peroxisomes. Auto is referring to self. No, don't think that. Auto is those phagosomes, those membrane, big structures surrounded within the cell. No, I don't think so. You have to check, and I don't agree with this. I rather still insist these are like what I told you. Yes, this is yes. Mitochondria is the only organelle which has a double membrane. So I think I have to have some break. You can come back for more chatting. So the class should be dismissed for a while. I think I have to leave the meeting now.